These are your second set of notes for plants, your photosynthesis notes, and the second set of 3 notes for the whole unit. So a lot of this is from middle school that you're familiar with, but we're going to cover it anyway just to refresh on it, and then we'll go into some more detail. Remember that during photosynthesis, plants, and it's not just plants, but all autotrophs are going to trap light energy and transform it into chemical energy, right? And it's not just plants, right? You actually have some photosynthetic bacteria that do this called cyanobacteria. You don't have to know this, but I'm just letting you, add it to your notes if you want. Cyanobacteria. And then some eukaryotes besides plants. So your protista, some protista can do this. Your plant-like protista can do this. We've seen Euglena has been able to do this, which is a unicellular eukaryotic protist. Algae are photosynthetic, so photosynthetic green-blue algae are all protista. They can also do photosynthesis, so it's not just plants. Sometimes we forget other kingdoms can do plants. So your protists and some bacteria can do it as well. And again, they're always taking light energy, so they're taking photons. Right in this little diagram down here, the energy that we're talking about it's not magical or anything like that. They're just photons, little packets of light, electromagnetic radiation, right? And they store it as glucose, so as chemical energy. So here's your overall equation. You'll recognize this from middle school. You don't have to balance it at all. But on the left here, these are going into the reaction. So these are your substrates. Remember, think back from Unit 2. Substrates are reactants. You have sunlight, CO2, and water. And then here are your products. Glucose and oxygen, right? And again, you don't have to balance this. And if you kind of look at the overall picture here, you can kind of see, where are the substrates? Here's energy, carbon dioxide from the atmosphere, water coming up from the roots via capillary action, right? And then you get your products out. You get glucose and you get oxygen released. So plants do this with a specific structure. Protists also do this with specific structures because they both have chloroplasts. Bacteria actually do this using their cell membrane. Remember, bacteria are prokaryotic. They don't have membrane-bound organelles. So they can still do this by using their membrane. And they also have chlorophyll and other pigments just kind of floating around in their cytoplasm. So you don't have to have the structure to do photosynthesis, but if you do have a chloroplast, it does kind of help make it more efficient and kind of keep the reactions to all the same area and not kind of all over the place like in a bacterial cell. So if you do have a chloroplast, this is the anatomy of it. It's a double membrane structure. You have an outer and an inner membrane. And it's kind of like a double membraned little miniature cell. So it has its own cytoplasm called the stroma. And we'll see this in a minute later. And you're going to see these little discs. They look like stacks of coins or stacks of pancakes, kind of all stacked on top of each other and connected to each other. These membranes, these little discs, are called thylakoids. And again, we'll see this more later. And the stack of thylakoids, the entire stack of membranes, is called a granum. And again, we'll see this in more detail later. So you do have to be familiar with the anatomy. Don't forget this. This is important stuff. That's why I'm pointing it out. So again, just as a reminder, chlorophyll is found in anything photosynthetic. If you are eukaryotic, like a plant or a protist, like algae, you have your chlorophyll in your chloroplasts. But if you are a bacteria, like your cyanobacteria here, then your chlorophyll just is loose, floating around in the cytoplasm. All right, so this reaction is a chemical reaction. It's mediated, it's run by, it's governed by enzymes, right? So all of these substrates are going to affect the rate of photosynthesis, right? The amount of sunlight you have is obviously going to affect the rate of photosynthesis. The more sunlight you have, the more photosynthesis you can do. Same thing with CO2. CO2 is literally the building block for glucose. This carbon from carbon dioxide is the carbon that's building glucose. So obviously, if you have some more CO2, you're going to be able to get more glucose, more substrate, more product. Similarly, the amount of water is going to help you regulate or increase the amount of photosynthesis that happens. And temperature is important because all of these enzymes have optimal temperature ranges. Too hot and the enzymes denature, too cool and the kinetic energy slows them down. So all of these are factors that dictate the rate or the pace or the amount of photosynthesis that's happening. So let's just look at some graphs. On the y-axis for all of these graphs is the rate of photosynthesis. So the higher up you are, the more photosynthesis you're doing. So this first graph here on the left is light intensity. How intense, how bright is that light? Again, since the light is a reactant, you need it to run this reaction. So the more light you have, the more photosynthesis you do, up until a certain point. It doesn't just keep going forever and ever. You'll notice that it does kind of plateau, it kind of flatlines. And that's just because it doesn't necessarily matter how much light you have if all of the enzymes are busy. If all of the machinery in the chloroplast is busy doing its job, those extra photons don't really do anything. You can only kind of go as fast as your slowest parts. If you think of this like a machine, and excess light isn't necessarily going to help after a certain point, right? It's a similar story for carbon dioxide, right? So again, the more carbon dioxide, the better, right? The more carbon dioxide, the more glucose you can make out of it. However, once all of the enzymes are busy, once all of the cell machinery is busy, excess carbon dioxide isn't necessarily going to help. So you do kind of, just like with light, you do kind of plateau and flatten off. And I know these two curves do look slightly different. That's kind of irrelevant. All you have to know is that they do increase up to a point where then they just kind of plateau because all of the enzymes are busy and there's simply nothing else that can help them go faster. And certainly not giving them more light or giving them more CO2. They're doing all they can. They're maxed out. Oops. All right, this last one you should be familiar with, right? Again, enzymes have an optimal temperature. Too hot, so 40 is past 100 degrees Fahrenheit, just for reference. Too hot and they do start to denature and their rate of photosynthesis plummets. And similarly, too cold, they don't denature per se, but they do slow down and bump into their substrates less, and that slows the reaction down as well. All right, so you guys are very familiar with the pigments, right? You've talked about chlorophyll in middle school all the time. These are molecules that absorb light. They absorb specific wavelengths, little packets of photons, little packets of energy. And they reject others, right? So this is why pigments will look certain colors. We'll talk about this in a second. But just so that you can look at it and are familiar with what a pigment is, this is chlorophyll A, this whole big molecule down here. You have this ring structure kind of on one side, and by the way, you don't have to memorize this. I'm just showing you just so you can appreciate it. You have this big kind of ring structure here on one side. You have what looks like a fatty acid tail. I wouldn't call it a fatty acid tail, but it is hydrophobic. It's just a bunch of hydrocarbons. But you have these CH3 groups that make it a little bit different from your typical fatty acid. So I wouldn't call it that, but it certainly is hydrophobic. If you look at this ring, what you're going to notice is that, hey, there's a magnesium in the middle. There's metal. There's literal metal. There's one magnesium atom. Magnesium is a metal in the center of this pigment. Where does that come from? Right? Where does that come from? If you go back here, well, it's not coming from sunlight. It's not coming from carbon dioxide or water. There's no magnesium in this reaction at all. Where is it coming from? It's actually coming from the roots. Right? This is one of the things, if you go back to the very, very first slide of the first set of content for this unit, we talked about how certain elements, certain atoms have to come from the roots because they can't come from anywhere else. So this would come from the roots. Just like nitrogen or phosphorus or sulfur would all come from the roots. Comes from roots. This is also why you need trace metals in your soil, because your plants need them to build chlorophyll. All right, there are a couple types of chlorophyll. Let's look at two of them really quick. This is called the absorption spectrum. So what you're going to see here is that on the y-axis, the higher up you go, the more light is absorbed. And then you have Roy G. Biv here on the x-axis, right? You have your red wavelengths over here, and you have your violet wavelengths kind of on the left. If you keep going, so if you went past 400, so if you went down to 300 nanometers, NM is nanometers, it's a distance, it's a really, really small distance. You have your, let's see, ultraviolet down here. And if you, this is your UV light, the kind that can give you cancer. And this is why you put sunscreen on, because this light can be very dangerous. And if you kind of go beyond 750, so like 800, 900 nanometers on the other side, then you get your infrared light, which is basically just heat. And then you've got, you know, all these other kinds of wavelengths that you can't see, but these pigments really only collect wavelengths that you can see, so we only focus on the visible light spectrum here. Anyway, so chlorophyll A and B do a pretty decent job. Look at this, up to 70, 85, 90% of all the wavelengths of light are getting absorbed by chlorophyll A and B. If you look at chlorophyll A, which is this blue line, it does a really good job, if you drop straight down to the x-axis, of absorbing, I don't know, wavelengths of light between 400 and 450, right? So it does a really good job at absorbing purple. Chlorophyll B does a pretty good job down here, but it does a really good job with about, let's say 475 to 500. And that corresponds to blue light. So violet, blue light gets absorbed by plants. Green wavelength light doesn't. Right, so right around here, right around 550 nanometers, which corresponds to what we would see as green light, chlorophyll just rejects that. It does not absorb that whatsoever. It does a little bit better with yellow and orange, and then it does second best, I would say, for both chlorophyll A and B, kind of around 650 to 700, and that corresponds with red light, which basically means chlorophyll likes purple, blue, red, some orange, and some yellow, but it really doesn't accept green wavelength light at all. It rejects green wavelength light. We just answered these. You can answer those on your own if you want. We also just answered that. And this is actually why plants will look green. The wavelength that you see is the wavelength getting bounced off the pigment, because the pigment is rejecting it. So what you see is what the pigment does not absorb. It's what the pigment is getting, is reflecting. So chlorophyll does not accept green light. It reflects green light. And because chlorophyll is the predominant pigment, it is the majority pigment in plants, plants tend to look green. And there is some variability on the species. Like some species are just more red or orange or yellow than others. But the vast majority of plants are going to look green because the vast majority of pigments are in fact chlorophyll A and chlorophyll B. Some of the other pigments though, again, it's not the only pigment, you have what are called carotenoids or carotenoids. This is from the word carrot, which is orange. So these are actually going to reflect or reject orangish red light. Xanthophylls look yellow. Anthocyanins, like a maple leaf, like the Canadian maple leaf on their flag, it's red because it has a lot of anthocyanins, so it rejects that red light. Some blue light also. So depending on the relative amount of these pigments, different species of plant leaves can look slightly different colors. And it all depends on just how much of these pigments are present. All right. So in the fall, so why do leaves change color in the fall? Leaves change color in the fall for two main reasons. The first reason is changes in light, right? What's called the photoperiod. You don't need to know this word, but I'll give it to you here. The photoperiod is the amount of daylight, right? How many hours of the day is the sun out? In the winter, in the fall and winter, the photoperiod decreases, which means there's less light, there's less reactant for photosynthesis. So plants respond. They say, oh, well, there's less light. Maybe I should do less photosynthesis then, right? It's a similar story with temperature. In the fall and the winter, the temperature also decreases. So you have these two factors that are directly related to the amount of photosynthesis and the rate of photosynthesis that both start to decrease. Plants, and this is an evolutionary pressure on plants, plants that didn't break down chlorophyll wasted precious resources and basically just went extinct. Plants that dropped their chlorophyll quicker or earlier in the year were able to survive and outcompete others. So evolutionarily, there's an advantage to actually breaking down chlorophyll and dropping the leaves and essentially going into a dormant state. And this is what you see in a lot of plant species and a lot of related groups of plants. They're called deciduous plants or deciduous leaves that drop their leaves in the fall and essentially kind of hibernate, if you will, during the winter. So chlorophyll literally breaks down. The plants stop building chlorophyll. They stop maintaining chlorophyll. Actually, a lot of plants store the chlorophyll in their trunks during the winter just because it is such an expensive, bulky molecule to make. And that magnesium is hard to come by, so you don't really want to destroy your chlorophyll. But you do kind of conserve it, you break it down, you put it back into the trunk, and the leaf turns brown, right? If you don't have the chlorophyll, if you lose that pigment, you're going to lose the green color. All right, well, your next question is, okay, well then why are the other pigments still there? Because this orange and this yellow is super pretty and the red is super pretty too. Well, chlorophyll is a little more sensitive. These other pigments that reflect orange, yellow, and red light last a little bit longer, right? And this is the key thing here. These other pigments are a little more tolerant of lower levels of light and a little more tolerant of lower temperatures. Not too much, but they can handle, another 3, 4, 5, 6, 7 weeks of it, right? So once chlorophyll has gone and that green color is not being reflected anymore, you can finally see the light being reflected from the other pigments that were in the leaf all along. And I think that's another interesting thing, is that it's not that chlorophyll was the only pigment there. It was just that chlorophyll was so powerful, it was so predominant, that you never had a chance to see the other pigments and the light they reflected until chlorophyll broke down first. So that's why leaves change color in the fall. It's a combination of the photoperiod decreasing and the temperature decreasing. Chlorophyll is the weakest pigment, so to speak. So it breaks down first. It gets stored and put away first. It gets locked up for winter. And the other pigments kind of have a moment to shine, quite literally. And then you see them, and then eventually even they get too expensive to maintain, and the plant just drops the leaf entirely, right? All right, so this is a little bit of extension. So this is what I call the honors extensions that I expect from you guys, just because you are honors and you do see a little bit more content. This would be a good pausing point if you want to take a break, because there's a little bit of a break here. So go ahead and pause if you want. All right, I'm assuming you've paused and you've come back. So here's the extra info. We're going to go into photosynthesis in a little bit more detail, and I need you to stay with me, and I'm going to do my best to get the big picture and simplify it. Photosynthesis has two main phases, two main stages, if you will. The light reactions, quote unquote light reactions, because they need sunlight to happen, and the dark reactions, also called the Calvin cycle, that does not need light, right? And we're going to see where they occur. So the light reactions occur in the grana of the chloroplast. Well, remember that the grana was this stack of thylakoids, right? The stack of thylakoids inside the chloroplast. Remember, we are in the chloroplast, not just the plant cell, in the chloroplast of the plant cell, right? The dark reactions, or the Calvin cycle, is going to occur in the stroma. That's basically like the chloroplast's cytoplasm, right? So let's look at them in turn. The light reactions, also called light dependent because they depend on light, again, they occur in the grana. And what's going to happen is water is going to get split. The hydrogens and the oxygen are going to get split from each other. All right? Enzymes are responsible for doing this, but right out of the gate, you get your first product. This is where all of the oxygen that you breathe from photosynthesis comes from. All of the oxygen you inhale and use for cell respiration originated from water molecules. That's where it comes from. That's pretty cool. And that is what's given off. That's the oxygen that you breathe. And all of the oxygen on Earth, in Earth's atmosphere that you breathe, is from photosynthesis. It's all from, there was no oxygen in Earth's atmosphere before photosynthesis evolved in bacteria. We're also going to make some ATP. We're going to make some ATP. Notice that this is a little bit different than what the mitochondria does. Yes, the mitochondria makes a lot of ATP, but it makes it for everything in the cell. The ATP being made here, we actually need to keep for the rest of photosynthesis. So technically, yes, ATP is made, but it's not really made for the rest of the cell. It's just for the next step in this process. All right, so here we go. The oxygen is released. The plant doesn't use it. Actually, some of it can go to the mitochondria, but most of it does get released. And then the hydrogen, notice we still have this hydrogen here left over, and the ATP we keep. The plant keeps it, and they're going to go on to the next reaction. And we'll return to this NADP plus in just a second. All right, so the next reaction, the light independent reactions or the dark cycle, they're all the same thing. The Calvin cycle, there's actually three different names for this cycle, but they all mean the same thing. Again, we're in the stroma. These do not require light, hence light independent or dark, right? Dark's kind of the bad word. It doesn't really make a lot of sense. Light independent is a lot better. And Calvin's the guy's last name who figured this out. We're going to take the hydrogen. I'm down here in the reaction. Here's the hydrogen from the light reactions that we just made from water. Here's the ATP that we just made. And we're going to take CO2. So this is where our last substrate's coming in. And we make our product. You make glucose as part of the Calvin cycle. You make glucose as part of the Calvin cycle. So let's wrap this all up, and I think this will help put it together. Here's your summary, right? So here's your chloroplast. Here's your double-membraned organelle. Your chloroplast has your two membranes. The light reactions are on the left. and the Calvin cycle, the light independent, the dark reaction is here on your right. So let's start. Here's light, your photons, and here's water. Notice that our third reactant, our third substrate is CO2, but we don't use that until the Calvin cycle. So we use light and we use water first. In the thylakoids, which is just the stack of grana, so I'm actually going to write that because that's what we've been talking about so far. So in the grana, that's plural, or in the granum, you have your light reactions. So light and water come in, and then oxygen leaves. So here's your first product. And I'll write reactant up here. All right. Here's the ATP that we said that we needed to make for the rest of the steps, right? So it's leaving the grana, it's leaving the thylakoids, the stack of thylakoids, and it's entering the stroma, right? This is all the stroma. All of this space out here is stroma, right? So it leaves the light reactions in the thylakoids and it enters the stroma. Here's the hydrogen right there. Remember we said that we made some hydrogen too? There it is. It's right there. It's on this molecule called NADP, right? NADP, or technically NADP plus, is like a school bus. It's like a school bus for hydrogen and electrons. It's a school bus for hydrogen and electrons. So what you're going to notice are these little blue balls with a little negative sign in them. These are electrons. That's supposed to be an electron, right? So this is a fully loaded school bus. Think of it just like that, or a taxi cab, or something like that, an Uber, right? It's taking the hydrogen and it's taking these electrons from the thylakoids out into the stroma where they're needed for the Calvin cycle, right? And if you back up, well, the hydrogen is going to get off that school bus because it's needed to build glucose. That's where the H comes from in glucose, right? So the hydrogen gets off the school bus, and actually so does the electron. So both the electrons and the hydrogen get off the school bus, and they return. They don't just hang out there forever. They return back to the light reactions in the thylakoids to pick up more hydrogens and more electrons. And that's why you have this plus sign here. And this is more AP Bio stuff than it is honors bio stuff. So you don't have to know this. But if you're wondering why there's a positive charge here, that's because the electrons are gone, right? And the electrons had a negative charge. Also notice that the H is gone, right? There's no H up here in the top anymore. It dropped the H off, right? Notice that we're also going to use some energy. So ATP is going to give the Calvin cycle energy, and it's going to return back to the light reactions to kind of start over. So it might be a little confusing now, but you will understand more about ATP and kind of what's happening here as we progress through the unit. And then also notice that here we go, we come out with sugar, right? So let's review it one more time. And if you understand this, you're golden here, right? The reactants are on the top, right? Light and water are used for the light reactions, whereas CO2 is used for the Calvin cycle, right? So here we go, step one. Light is used to help break down basically water with the help of enzymes, right? So the oxygen in water leaves. That's a product. The hydrogens load onto this school bus, right? Here's the empty school bus coming in from the Calvin cycle. Empty school bus. The hydrogens from water load onto it, as well as some electrons, and then they leave. See, it's a fully loaded school bus down here. They leave to go off to the Calvin cycle, right? They're dropped off at the Calvin cycle, and then the empty school bus comes back to pick up some more, and over and over and over again, right? They load up, right? The light reaction is the bus stop. They load up here at the light reaction, they get dropped off at the Calvin cycle. The empty school bus goes back to the bus stop, et cetera, et cetera, et cetera, right? It's an endless cycle. Once you're in the Calvin cycle, CO2 comes in, and that's how you make your sugar. And you don't have to know any of the details. That's AP Bio stuff. But if you get the basics, again, you're good to go. We will review this in more detail in class. I don't expect you necessarily to get this all at once, and I do know these notes were kind of long, so I do want to spend a little bit more time talking about it together. But I do hope this helped, and I will see you later.